So, in this lecture, we're going to look at these ectodermal and neural crest cells, and specifically their role in the formation of the vertebrate head. What are we going to do in this lecture? Well, there'll be a little bit of revision of the last lecture, and we'll talk about neurulation and how the neural crest cells form from neurulation and their role. We're going to look at the neural crest cells and how they are derived from both the head and the trunk, and how these different cells undergo different fates. And then finally, we're going to look at developmental similarities between these neural crest cells and embryonic stem cells. So remember, a process of neurulation where we get the closure of the neural tube and its separation from the epidermis. Well, the last part that separates from the epidermis are those neural crest cells. So this is in primary neurulation. These neural crest cells migrate away to form the crest of the invaginating neural tube to form a wide variety of peripheral tissues. So here we have our neural tube, and on top of that, forming a crest, are these neural crest cells. So where do these cells go? What do they form? Well, people have performed what are called fate mapping experiments, where you label the neural crest cells either with a fluorescent dye, or you genetically label them with a marker for a gene you know is expressed in neural crest. And then you look to see where these cells end up, how they migrate through the developing embryo. And this fate mapping process, either with dye or by a genetic method, either electroporation of a marker gene or by transgenic knock-in of a marker, revealed that there are two key migratory routes. There's the melanocyte route, which is the late route, and this is our red cells migrating from the dorsal root ganglion to form melanocytes. Or there's the early route, the peripheral nervous system route, where neural crest cells give rise to either neurons or peripheral oligodendrocytes, Schwann cells. So there are these two key migratory routes. So it's weird that the ectodermal cells become neural crest cells and then migrate back into the epidermis in the case of the melanocyte. So let's look at neural crest fates and how these relate to the formation of the head and the skull. So our undifferentiated neural crest cell can, under the control of a number of different transcription factors, adopt a number of different fates. So typically neural crest cells, because they're neurally derived, will either express SOX9 or SOX10. And then different factors after the expression of these key transcription factors will differentiate the neural crest cell, causing it to undergo one of several fates. If they express the melanocyte-specific genes MITF, DCT and TRP2, which are actually pigment-forming cells, these cells will pigment-forming proteins. Then these cells will become melanocytes. And you can see different combinations of transcription factors will lead to different fates, such as Schwann cells, sensory neurons of the central nervous system, and neurons of the autonomic nervous system. Only if these cells express SOX9 and then express collagen 2 alpha 1 do we get cartilage derivatives, so things that make cartilage as part of the skull from these neural crest cells. So neural crest cells only form from so the ectomesenchyme, which are these bone and cartilage cells, only form from cranial neural crest, so neural crest from the head. And so we have all these diverse fates of neural crest cells, and this largely depends on the local systems that these neural crest cells receive in different parts of the body. And there's a clear distinction between the fates in the head, the ectomesenchymal fates, and the fates in the trunk, the non-ectomesenchyme. So our ectomesenchymal fates include the formation of hindbrain, the midbrain, and the forebrain structures, so the bone structures around these tissues, whereas the non-ectomesenchyme, dependent on where we are, forms dorsal root ganglia or the adrenal gland, and various innervations of tissues associated with that region. So neural crest cells will become the gut enteric ganglia and innervate the gut. And so different parts of the embryo drive these neural crest cells to different fates. In the head itself, there are a number of discernible neural crest streams that are seen during head development, and these form the structures of what's called the frontonasal process, so it's this region here of the developing skull, and the pharyngeal arches, which are these things that lie behind the frontonasal process. And these are the main streams of neural crest migration in the head structures. So these neural crest migration patterns are quite complex. They require the down-regulation and up-regulation of certain proteins to, once these cells have migrated to these regions, to adapt the cranial neural crest fates. So the migratory cells down-regulate neuroglial markers such as SOX10 and FOXD3, and they then express fibroblast growth factors and this homeobox gene called DLX, and this specifies those structures the neural crest are forming in the developing skull. So here we have a schematic of the formation of the face and the migration of these processes to form the structures. So we have our frontal nasal process, our maxillomandibular process, and our lateral nasal process, and you can see here how they contribute to the different parts of the developing head. The frontal nasal process contributing to the upper head and parts of the nose, the lateral nasal process contributing to the lateral part of the developing nose, and the maxillomandibular process contributing to the jaws of the skull, of the face. And you can see how these processes form the various parts of the skull and the head in this movie. You can see the movement of the frontal nasal process in red, the lateral processes in yellow, and the maxillomandibular process in blue. So obviously, if things happen to prevent the proper migration of these cells, the defects in developing humans would be quite severe facial defects. So the formation of the face is quite complex in so much as the neural crest cells, the cranial neural crest cells, only form some of the bones and cartilage in the face. And these pharyngeal arches, 1, 2, 3 and 4, undergo complex morphogenesis to form the face. So pharyngeal arch 1 contributes to the malleus and incus of the ear and also the Meckel's cartilage in the jaw. We have pharyngeal arch 2 contributing to the stapes and the styloid process of the ear and the hyaloid cartilage. We have pharyngeal arch 3 also contributing to the hyoid cartilage. And then we have pharyngeal arch 4 that contributes to the laryngeal cartilage. And then other tissues contribute to facial bones and the rest of the skull. So you can see here that from those initial pharyngeal arches, there's complex morphogenetic processes that form the face. So, as I alluded to earlier, the cranial neural crest cells and their inability to migrate or form the correct tissues underlie many facial developmental defects. So if you have defective neural crest migration, cranial neural crest cell migration, we get two diseases called Treacher-Collins syndrome and oral facial syndrome. You can also have defective facial mesenchyme morphogenesis, and this can lead to cleft lip and palate, which is the most common craniofacial defect in newborns, and also FGF hyperactivity syndromes. Such as Apert syndrome, as we see here. And down the bottom panel here is, we can see the defective patterning of the teeth here in this oral facial digital syndrome, which is caused by incorrect migration of these neural crest cells. In extreme cases where we get defective facial mesenchyme morphogenesis, we can get holoprosencephaly in a similar manner to when Sonic Hedgehog is inactive during development. So, as we had mentioned before, facial bones don't all come from the neural crest cells. Some of it is also mesodermally derived, and that's not surprising because bone is a mesodermal tissue. We can see in developing mouse skull where the contribution to each of these tissues arises, and we can do this using specific Cre recombinase expressing marker mice under the control of particular promoters. So Wnt1 is expressed in the neural crest, and LAC-Z is a reporter gene that encodes beta-galactosidase, and then when you add a reagent, you can see a blue coloration. So here, with this Wnt1 Cre, we're looking at the neural crest-derived bone, and we can see where there's blue. We can see these are the regions where the neural crest derives, is the source of the bone. MESP is a mesodermal marker, and if we do the same experiment but use an MESP Cre expressing mouse, we can see in effect the inverse of the Wnt1 Cre. So we can see here in blue what bones are formed by the mesoderm, and you can see that gap there indicated by the star, which is where we have a little spot of Wnt1 Cre positivity, so neural crest derived bones. We see this gap here, so there's a very complex combination of bones with different developmental origins in the cranium. So here we have a schematic of all of the facial bones in the head and what ones are formed by mesoderm and what ones are formed by the neural crest. And as you can see, the majority are formed by the neural crest, but there's that small percentage, the parietal, temporal optic, etc., are produced by the neural crest, and that interparietal bone, that little gap, that star we saw in the previous slide, is produced by neural crest cells. So why can cranial neural crest cells form ectomesenchyme and trunk neural crest cells can't? This is to do with the specific expression of this protein SOX9. Neural crest cells can only become ectomesenchyme when they express SOX9 and then express collagen 2A1. So how do we produce this ectomesenchyme? It's not just the expression of SOX10. And we can show this in this experiment here, where we use this drug SU5402 to inhibit FGF signaling, inhibits fibroblast growth factor signaling. And we'll look at that in a later lecture. But suffice to say that if you inhibit FGF signaling, we lose markers of ectomesenchyme. So DLX2, remember, is that marker that specifies that neural crest cells become ectomesenchyme. And so we can see here, the blue marker here is a marker of expression of DLX2 or SOX10. So we see here SOX10 expression in the pharyngeal arches. Here we see SOX10 expression still in the presence of our drug inhibitor, so FGF signaling doesn't prevent that initial specification. But the expression of DLX2, this is shown here in our control, our DMSO control, is absent from our treated embryos. So FGF is absolutely required for the formation of ectomesenchyme. And this is an experiment in zebrafish, but it works the same in other organisms. So no DLX2, no ectomesenchyme. And we already showed that Wnt1 targets the neural crest. So this is a marker of the neural crest and also the dorsal neural tube. So you can use a Wnt1 Cre, where it's expressed as a reporter gene under the control of that Cre. You can use Cre to activate the reporter. And we can see here that there's expression in the neural crest along the whole length of the organism. So we can use these Wnt1 Cre mice to knock out expression of other genes. So in this case, we're knocking out the BMP receptor, TGF-beta 2, only in cells that are expressing Wnt1. So it's only the Wnt1 expressing cells that have Cre, and therefore you only get removal of TGF-beta 2 when Wnt1 is present. And we can see these skeletal preps of control and mutant mouse skulls. And in this stain, blue is cartilage and red is bone. And so we can see in our mutant here, the lack of BMP signaling leads to a almost complete absence of bone formation. Our bone derivatives of the head are lost, but cartilage, which is expressed here in blue, which is stained in blue, is present and normal. And this phenotype is a disease called the George syndrome, a genetic disease, an autosomal dominant birth defect that occurs in 1 in 4,000 births due to a mutation in this TGF-beta 2 receptor. If we then do, if you like, the inverse experiment and get rid of SOX9 in these Wnt1 crees, we get complete lack of neural crest-derived cartilage and endochronal bone. So here we have the same stain, our same pink and blue stain. Blue is bone, pink is cartilage, and we lose specifically the cartilage and bone that comes from the cranial neural crest cells. So now we have this model of ectomesenchymal specification, by which we have multipotent neural crest stem cells, and through a process of expression of transcription factors, inhibition of the expression of other transcription factors, and activation of fibroblast growth factor signaling, we can make these neural crest multipotent stem cells go towards an ectomesenchymal fate. So these ectomesenchymal progenitors represent a stage of development beyond the neuroglial progenitors. However, they are only found in the cranial region and only form a subset of bones, those that are developmentally related to cartilage, so-called endochronal bone. This happens because of the downregulation of FOXD3 and SOX10 and that upregulation of SOX9. Remember that if we don't have SOX9, we lose the neural crest derived cartilage and endochronal bone. So SOX9 is required to form these ectomesenchymal stem cells that are able to produce bone and cartilage. So the neural crest actually is quite an interesting cell. It shares many capabilities with the blastula, and this is weird because it doesn't really fit into this concept of progressive specification that was the idea of Waddington's developmental landscape. So you can imagine a ball rolling down a hill, and it will take a path dependent on what ridges it encounters. So it encounters this first ridge and it can either go this way or this way, and then it encounters a second ridge. It can either go this way, and this way, and this way, or this way. So this ball will roll down the hill. This represents the fates that the cell undergoes, and it can undergo an epidermal fate, a neural fate, a mesodermal fate, or an endodermal fate, like all pluripotent cells can. So where do neural crest cells fit in this scheme? Well, shared regulatory programs between neural crest cells and blastula stage cells suggests the retention of blastula stage potential in neural crest cells. This suggests that they have some of the capabilities of blastulas to form all cell types. So in effect, they have the potential of this one cell division cell. They can be all cells, or all kinds of cell fates. And so how can you look at that? So these people looked at how the neural crest shares gene expression with the blastula. And how was this done? Well, here we have our animal aspect and our lateral aspect. We're doing an in situ hybridization for genes that are blastula markers and genes that are neural crest markers. And so you can see here in these blastula stage embryos that not only are blastula markers, bona fide blastula markers expressed, but also neural crest markers expressed very early on during the development of this embryo. So we have ID3, we have SNAIL1 required for that mesenchymal to epithelial transition. We have FOXD3 that's required for neural crest cell specification towards an ectodermal fate. So you can see here that blastulas express these cells in the animal part of their embryo. So they express them very early on in development. And what's interesting is if you remove these neural crest markers, such as SNAIL or SOX5, this can actually reduce the pluripotency of these cells. So it causes a loss of pluripotency markers. So here is our dominant negative SNAIL that we've added. It's a SNAIL that doesn't function as a transcription factor. And here we have the knockdown of another neural crest cell, SOX5, by morpholino. And in both cases, wherever we see a knockdown of SNAIL, we see loss of these pluripotency markers. And we see that in both cases for things like ID3 and TFAP2. So removal of SNAIL or SOX5 transcription factor activity causes loss of these pluripotency markers. And in later stages, the ability to differentiate requires these neural crest genes. So Brachyury is a mesodermal marker, and in our normal embryos we see normal formation of expression of brachyury. When we knock out this key neural crest marker, SNAIL, we see a loss of mesodermal fates. So this suggests that the neural crest is ectoderm, is not ectoderm and requires the ability to form mesoderm. It always has this ability to form mesoderm, and this holds true for more developed mesodermal tissue such as muscle. Remember, the transcription factor MyoD specifies muscle. We can see muscle expression in our Xenopus embryos, and in our delta snail embryos we don't see MyoD. But it seems very important to point out that these two figures are duplicated. I think this was just an oversight, but and not deliberate, but this is the same picture. So just be aware when you're reading papers to see whether there are any weird inaccuracies like this. I'm sure they weren't trying to obfuscate things, and I'm sure that if you have no Brachyury, you're not going to have any MyoD. But to recap, at later stages, the ability to differentiate into mesoderm requires these neural crest genes. So, because neural crest genes share gene expression with blastula cells, the old idea that neural crest cells were ectodermal then became neural crest cells, and then adopted the mesodermal endodermal fates is somewhat wrong potentially. The neural crest evolved, according to the authors of those papers, by somehow maintaining blastular-like abilities, i.e. they're pluripotent cells. So our speculation, based on what we see, is that our neural crest cells are basically pluripotent stem cells and have the ability to form epidermis, neural cells, mesodermal cells, and endodermal cells. And so the final kind of cell that we have in our neural crest are these olfactory ensheathing cells. Now, if we express SOX10, which we know marks neural crest cells throughout the body, except for the ones that form the skull, we have a SOX10 LAC-Z mouse, and we can see all the neural crest cells expressed and their presence in the embryo. We can see all the things that aren't SOX9 required, so none of the skull, but we still see olfactory cells. So some olfactory cells are neural crest cells, and then we can see that the heart arches have neural crest cells. We have dorsal root ganglia. So this is everything else. But you can see here that even the SOX10 cells contribute to some of the cells that are important in the head. In the olfactory system, these Schwann cells are known as olfactory ensheathing cells, and they are constantly produced throughout life. So now we've looked at all the different kinds of neural crest cells and how even the SOX10 expressing ones can contribute to head-related fates. And here is the region in that olfactory pacode where these ensheathing cells are expressed, or are present rather, and produced constantly. So that's the end of this lecture. What did we look at? We looked at revision, and we looked at the process of neurulation and the formation of the neural tube. Then we looked at how that produces the neural crest cells. At the edge of the neural plate, we get neural crest cells that then sit on top of the neural tube, and then these can migrate to a number of tissues in two waves of migration. The first wave being a primary wave where they migrate and form many of the dorsal root ganglia and tissues of the head. And then we have that secondary route where we produce the melanocytes. We saw that difference between SOX9 and SOX10, where SOX9 produces neural crest cells that form the structures of the head, and then SOX10, which forms all the trunk derivatives of neural crest cells. We looked at other processes that are required, the expression of ectomesenchyme that's required to make parts of the skull, and the requirement of FGF signaling and the homeobox factor DLX2 to make cartilage, and the BMP requirement to make bone. And then lastly, we looked at key similarities between neural crest cells and embryonic stem cells, or stem cells derived from the blastula, and how neural crest cells may continually have the capability to form all the three different germ layers, so the ectoderm, mesoderm, and endoderm. So I'm going to stop there. Rest, relax. We have office hour at the normal time, and I will see you there to ask your questions. All right, see you later. Bye.